The first part of interphase is the G1 or gap 1. This is where the growth of a cell takes place before DNA is duplicated. The second part of interphase, the S phase or synthesis, is when DNA is duplicated. This is crucial to ensure that both new cells have a complete set of DNA. The last part of interphase, the G2 or gap 2, prepares for cell division. This is when DNA replication is checked over and any errors that may have occurred are fixed. The first stage of mitosis, called prophase, is when the DNA begins to condense. You can see here that new microtubules are assembling, and then they will move to the centrioles at opposite ends of the cell. You can also see here that the nuclear envelope is beginning to break up. The next part of mitosis, called metaphase, is when the chromosomes align at the equator. At this point, they are in their most tightly condensed form. A way to remember metaphase equals middle is that metaphase and middle both begin with an M. The third part of mitosis, called anaphase, is when the two sister chromatids break apart. They are now each separate chromosomes. The microtubules pull them to opposite poles. One way to remember anaphase is that it equals apart. Both of these start with an A. The final stage of mitosis, called telophase, is when each new cluster of chromosomes begins to decondense. A new nuclear envelope begins to form, and mitosis is complete. The final stage of cell division is called cytokinesis. This is when the cytoplasm splits. The plasma membrane begins to cleave until it pinches into two new cells. Cell division is now complete.